So we've been talking about properties of water, and this last little section on pH I rushed through a little bit, so I wanted to spend a little bit more time on it. So for AP Bio, you don't actually have to calculate pHs, you won't have to plug logs into your calculator or anything like that, but you do need to have a general understanding of how pH works. So it is a measure of the strength of an acid or base. It's the negative log of the hydrogen concentration. The little brackets around a hydrogen mean concentration. Actually, brackets around anything in chemistry means concentration. So here's the thing. You won't need to actually take logs because they use whole number pHs on the AP exam to make it simple if you're getting them from a hydrogen concentration. So if the hydrogen concentration was 10 to the negative 7th, if you take the negative log of that, the pH becomes 7. And if it was 10 to the negative 4th, then the pH would become 4. For water, it's 7 because the hydrogen and the hydroxide are equal to each other. But the part that throws people off is this, understanding, because it is a negative log, understanding how pH relates to hydrogen concentration. So again, I mentioned this in the last lecture, if the pH goes down by 1, from 7 to 6, or 6 to 5, 2 to 1, the hydrogen concentration goes up by 10 times. And in the same vein, if pH goes up, so let's say the pH goes from 9 to 10, then the hydrogen concentration would go down 10 times. That also means that if the pH, let's say the pH went from 5 to 9, how much would the hydrogen concentration change? So you'd say, okay, 5, 6, 7, 8, 9, right? If you count the jumps, it would go 10 times, 10 times, 10 times, 10 times. So 10 times 10 is 100, times 10 is 1,000, times 10 is 10,000. It would go down, because the pH is going up, the hydrogen concentration would be going down 10,000 times. So that's sort of how you would calculate pH. The thing that throws people off, if they've never done pH before, is the fact that they're inverse, that the pH going down means the hydrogen concentration is going up, and the pH up is the hydrogen concentration going down. And the other thing about that, the reason you need to understand that is because we're going to talk in a second about what are called buffers. So pH below 7 is acid, above 7 is base, and exactly 7 is neutral. And the reason why 7 is neutral is because, if you remember, originally I said that water breaks up into hydrogen and hydroxide. And in water, in a mole of water, the amount of hydrogens would be 10 to the negative 7th, and the hydroxides would be 10 to the negative 7th. So a very tiny amount. But what's the negative log of 10 to the negative 7th? 7. And that's why the pH of water is exactly 7. So it goes from 1 to 14, generally. And here's just some examples of some different pHs. So you can see acids below 7, so coffee's acidic, orange juice, vinegar. Acids usually taste sour. That's one of the characteristics. Bases often taste bitter, actually. Baking soda, ammonia, bleach, drain cleaner, soap. They feel slippery a lot of times, bases. That's because they're actually dissolving the fat in your skin, which is not a good thing. Okay, so buffers. Buffers are actually kind of complicated, but for AP Bio, here's the main thing you need to know. A buffer is just something, in living things in particular, that prevents pH changes. So here's again where people get thrown off. So if a solution was starting to become acidic, what would that mean? If a solution, let's say we wanted it to be neutral, but something was happening in our body that was causing it to become acidic. That would mean that the pH was going down, and it would also mean that the hydrogen concentration was going up. So what a buffer could do is a buffer would basically pick up the extra hydrogens. What's that going to do? Well, if it picks up the extra hydrogens, it's going to counteract the acidic solution and bring it back towards neutral again. Now, it doesn't always have to go to neutral, but that's the idea. So what if it was becoming basic? What if something was happening in your body that was causing the pH of your blood to become basic or alkaline? What would that mean? Well, that would mean that your pH was going down, and that your pH was going up, and your hydrogen concentration was going down. pH up, hydrogen concentration down. That means your hydrogen concentration is getting too low. So what would the buffer do? The buffer could add hydrogen ions to counteract it. So a buffer is sort of pushing the solution back towards what it originally was. A good buffer example is hydrogen carbonate. So it can either become H2CO3, it can break into HCO3, you don't have to know this buffer, plus hydrogen. So if we think about it, what's going to happen if our blood's becoming too acidic? Well, we have too many hydrogens. So what can happen is the buffer will pick up the hydrogens and shift this way and pick up those hydrogens, and now the hydrogens concentration drops in the blood because the hydrogens are now being carried by the hydrogen carbonate. On the other hand, if our pH is getting too low, this hydrogen carbonate can give away its hydrogens and put them back into the solution. So imagine a solution and it has a certain number of hydrogen ions. And what the buffer can either do is if somebody adds an extra hydrogen ion, the buffer can pick it up. If there's not enough, the buffer can add some. So that's how buffers can work to moderate pHs and keep them stable. Again, it doesn't necessarily mean that it keeps the pH exactly at 7, but it can keep it from changing. Again, the biggest thing is people get it backwards. So remember, if something's becoming acidic, that means you're starting to add hydrogens. The hydrogen ion concentration is going up. What would the buffer do to fix that? Grab some of those hydrogens and carry them away. So it goes from HCO3, it grabs a hydrogen, now the hydrogen concentration went back down. On the other hand, if there's not enough hydrogens, the H2CO3 can get rid of a hydrogen, become HCO3, and now it added a hydrogen ion. So it's going to release or pick up the hydrogen ions. In your body, you don't have to know this, I wrote not on the test, but they may mention on the AP exam buffers. Your body has all kinds of buffer systems to protect you. So a bicarbonate system, this is in your blood. There's a phosphate system. There's a protein buffer system. There's a respiratory mechanism. You can change how quickly you breathe because carbon dioxide also becomes hydrogen carbonate or H2CO3. So carbon dioxide levels can affect hydrogens or acidity and alkalinity. And renal, that's your kidneys. So you can actually pee out extra hydrogen ions. So there's a lot of things that your body would do to prevent your pH from going too low or too high and maintain homeostasis.